Hello, my name is Natalia Della Riva Herrera, and today I am presenting my recommendation on improving laboratory sterility and learning conditions in UTSA's undergraduate biology teaching labs. My goal is to show how current lab conditions affect scientific accuracy and student learning, and what changes would most effectively strengthen UTSA's reputation as a growing R1 institution. Why laboratory conditions matter. So, hands-on laboratory experience is essential for biology and pre-med students. Accurate experiments depend on sterile conditions, functioning equipment, stable temperatures, and reliable supplies. However, UTSA students consistently report preventable issues in the main campus teaching labs, including malfunctioning microscopes, contaminated workbenches, unstable temperatures that affect bacterial cultures, and shortages of sterile supplies such as pipette tips and gloves. My guiding research question for this project is, to what extent do UTSA's current laboratory conditions support accurate, high-quality scientific learning, and what improvements are most needed to reduce contamination and equipment-related errors? Identified issues in UTSA biology labs. From student feedback and my own experience, several recurring issues stand out in our biology teaching labs. These include malfunctioning microscopes, incubators, and water baths, inconsistent sterilization of shared surfaces, temperature fluctuations that affect cultures, shortages or expired supplies, and overcrowded bench space during busy lab sections. Together, these problems undermine both experimental accuracy and student confidence. How the data was collected. To understand how widespread these problems are, I surveyed 41 UTSA students who have taken undergraduate biology labs, ranging from freshmen to alumni. The survey included Likert scale, multiple choice, and open-ended questions about equipment reliability, cleanliness, temperature control, and supplies. I then compared their responses with published research on laboratory quality and contamination risks to see how UTSA's teaching labs align with broader standards. Equipment malfunction among students. The first visual illustrates how many students experienced equipment malfunctions during a lab experiment. In my survey of 41 UTSA biology students, 58.6 reported experiencing equipment malfunctions, most commonly in microscopes, incubators, and water baths. This means more than half of students encounter unreliable tools that directly undermine the accuracy of their results. The second chart shows how many students had to repeat experiments due to contamination, supply issues, or environmental factors such as temperature and sterility. 56.1% of students said they had to redo at least one experiment for these reasons, compared to 43.9% who did not. Repeating experiments for preventable reasons delays learning, reduces motivation, and makes it harder for students to trust their own results. Supportive evidence from research. These results match national findings. Research shows that inadequate sterilization, outdated equipment, and poor temperature regulation significantly increase contamination risk and reduce experimental reliability. Students also reported issues such as unclean microscopes, bench residue, expired materials, and overcrowded workstations. Together, the qualitative and quantitative data confirm that current conditions do not consistently support accurate scientific learning. To improve accuracy and reduce contamination, I recommend strengthening sterilization practices, exceptionally consistent cleaning of microscopes, benches, and shared tools. UTSA should also repair or replace outdated equipment and improve temperature control in lab rooms to protect sensitive samples. Better inventory management is needed to prevent shortages of sterile supplies, and brief student training on proper handling and cleanup can significantly reduce cross-contamination. If these changes are implemented, we can expect several positive outcomes. Students will achieve more accurate results and fewer repeated experiments, thereby strengthening their scientific skills and preparing them for STEM and medical careers. UTSA will waste fewer materials and lab hours on preventable failures, and overall, our teaching labs will better align with the standards expected as a growing R1 research institution. In closing, the evidence clearly shows that UTSA biology students face recurring laboratory issues that impact learning accuracy and scientific reliability. By investing in sterility, equipment reliability, temperature stability, and supply consistency, UTSA can significantly improve student outcomes and better align with R1 institutional standards. Thank you guys so much for listening today. I'm happy to answer any questions that you guys may have.